Okay, good morning to all of you. So today is our lecture. It is lecture number 12. It is based on our topic number 4, and we have seen the 4 points: sources of data for the systematics as morphology, anatomy, cytology, and embryology. Then fifth point is there. It is the phytochemistry as a source of data for the systematics. I think you have gone through this firstly. So give some examples and the importance of this. So what is phytochemistry? So it is the occurrence and distribution of various types of chemicals present in the plants. Right. So we are knowing different plants and the plant parts. They are having different chemicals, and this study their study with reference to the occurrence and distribution. It is generally related with the phytochemistry. The secondary metabolites like alkaloids, terpenes, glycosides, glucosides, phenolics, etc. So they are to be useful for the taxonomic references or the evidences are there. Nowadays plant classification is based on the particularly chemical classification, so it is having the importance generally in the recent classification, but some pharmacological studies are there, different Ayurvedic preparations are there, so they are based on this, or chemical preparations, they are based on the plant chemical nature. So therefore it is to be considered as a by the principle, the procedure, result, and the investigation. So from the each plant it carries the weightage. There are the two purposes of classification and the knowledge of the phylogeny. So what is phylogeny? Already we have seen the similarities and the differences. So therefore for classification this chemical nature, it is very important. Initially there is having the one point is there, as you are observing in the 19th century the chemical information of herbals, physiological activity, techniques, developments, it is to be studied, in which the saponins and alkaloids having the weightage. The chemically biosynthetic pathways develop into the micro and macro molecules metabolites. So different minor and major, there are the molecules are there, so they are to be studied. The micromolecules are mainly divided into primary and secondary metabolites. The primary metabolites involve the phytal pathway, which is secreted with the leaf extract, right? So for example, production of citric acid, proteins, amino acids, generally common seen in the grass, Gligiocolia. Gligiocolia is the good example. So you remember this. Then secondly, it involves in metabolic pathway. Example, proline in the seed extracts are there, or aspergin in the flower extract is there. So these are the primary. Then what are secondary metabolites? So these metabolites involve non-vital byproduct compounds, for example phenolics, alkaloids, terpenes. We are knowing some plants having a specific odor, so it is due to this particularly content of non-vital byproducts. So they help in defense against the prediction, or to refuse the pathogens or allelopathic agents, or help in pollination, or the dispersal of the seeds are there. So we are knowing some plants are having the specific secondary metabolites, so they are generally good character against the predation. आपण पाहतो की काही प्लांट्सच्या sheep म्हणा, goat म्हणा, they are not eating. Why? Because they have very strong odor is there. Next one important point is there, as you are knowing there is having these macromolecules divided into cementide and the non-cementide. So we have seen the micromolecules in primary and secondary. So now these macromolecules, cementide means which having the information of carrying molecules, for example DNA and the RNA. which and the protein, and non-cementide not involved in the information transfer, for example starch and the cellulose. So these are the major two types are there. The good examples for these, the phenolics having the characteristic smell in the catechol or the hydroquinone or the pyrogallol is there. Secondly, flavonoids are there, isoflavones and the malvadines or anthocyanin and the anthoxanthins, beta cyanins and beta xanthins are there. So you are knowing the good flavonoids as a anthocyanin, different colored petals are there. So these are nothing but the different anthocyanin pigments are there. So you remember this cementide and the non-cementide. Cementides are generally good example DNA, RNA, which are present there, and in the non-cementide carbohydrates and the starches are there. And then alkaloids are having the same example: nicotine in the tobacco, nicotiana tabacum; ephedrine, ephedrastane, it is a gymnosperm; then morphine, opium poppy capsule; latex, it is the latex of papaver somniferum. Papaver somniferum म्हणजे आपण जी खसखस वापरतो. So these are the seeds of this opium poppy capsule. Capsule म्हणजे fruit, unripe fruit, right? So remember this morphine. But त्याचा latex जो असतो त्याच्यापासून narcotic drug तयार करतात. Then tropane alkaloid from the Solanacea member and convolvulaceae. Solanacea म्हणजे धतुरा वगैरे आपण बघतो. We having the specific odor is there, right? Then fourth example about the terpenes are there. So you are knowing polymerized, these are polymerized isopropane derivatives. Good example mentha. Mentha म्हणजे पुदिना. पुदिनाचा aroma आपल्याला माहिती आहे, right? So you just remember all these examples. Then example number 5 about the amino acids are there. These are the peptide chains of amino acids, and proteins are with nucleic acid called the cementide. That is, DNA is primary cementide and RNA is the secondary cementide. So like this, as we have some good examples are there. Recently you are listening the good word as a nanobiotechnology. Nano means very smaller. So it is in the microns. So it is hybrid discipline, that is combination of biotechnology and the nanoscience. Nanoparticle size is below 100 micron, applied in the several fields of biosciences, right? So very 100 micron is equal to 1000 micron is equal to 1 mm. So just to see about the this parameter. Then it is applied in various fields nowadays of biosciences and biomedicine in commercial application. So nowadays there are the specific drugs available on the each and every disease due to the their activity, and therefore these are directly separated, detected from the plants are there. So therefore it is tools for the pathogen bio detection, then tissue engineering, MRI contrast enhancement. Then there is having the use of drugs, they impiling the direct movement of the nanoparticles to specific organ, or the organs are there. So what happens, we are knowing allelopathy, chemical medicines are there. So firstly they goes to the digestion, that run to the into the blood, and through arteries again it is given to the specific part, right? So therefore the nano biotechnology is an important tool, and therefore it having the importance by phytochemistry as a source of data. So in taxonomical condition, so it is for the study of phylogeny, similarity and differences in data of nucleic acid and gene level change. So we may do the thousands of plants, hundreds of plants for the study, and therefore this is the one part is there very nicely. Then secondly, as you are knowing, the molecular evolution data generally from the plants, plant physiology, biochemistry, anatomy, palynology and the embryology. Palynology means the study of the pollen grains are there, so it is to be studied. Then it gives the similarity and in the genetic material, and it differences in the nucleotide sequence and the changes in the DNA. So it is very important to study, and therefore just you give what is the role of the phytochemistry in the system. Second good example is there, as it gives the location of molecular data. There are 3 locations in chloroplast and mitochondria. So right, so inherited by maternally, nucleus inherited by the parentally. So what is this? So chloroplast, 3 different genomes are there. So different genomes. Chloroplast genome, it is smallest, 120 to 160 kbp, kilobas pairs, right, which are highly stable. Then secondly, mitochondrial, 200 to 2500 kbp. Then nuclear, this mitochondrial undergoes lot of rearrangements in the cell, and nuclear it undergoes the millions of the rearrangement, which is the highly stable. So you remember this second one, molecular data. And thirdly, molecular systematics are there. We are knowing the molecular techniques. We are observing next point. So innovation in the new whole DNA. Nowadays genetic engineering, it is related with the DNA of the each and every plant. So breakup of DNA at the sequence and multi-copying of the polymerase chain reaction, commonly it is called as PCR, polymerase chain reaction, helps in the data generating. For example, grass genomes analysis done, so in rice, maize and the wheat. So in rice 4000 kbp are genomes, then in maize 2500, and in largest in the wheat 17,000 kbp. So you refer this. And last point about this, according to Mendel and others are there in 1995, so they have innovated the new world tetraploid cotton, right, Gossypium species. So Gossypium hirsutum, which is formed by the allylopolyploidy of the genomes from old crossed with the new world cotton. So this allylopolyploid developed, it is from the old cotton and the new world cotton, and where the old cotton chloroplast is from the African cotton, which having about 1 to 2 million years ago history. So this is our today's lecture. We will see about the next last point, molecular data as a source of source in the taxonomy or in the systematics. So thanks to all, have a nice day.